Staining is going to aid us in the visualization of bacterial cells. Bacteria are very small and very hard to see because they have the same refractive index as water. So staining helps us see them better. Also, we can determine their cellular morphology or shape, and this is an important part of their identification. There are three basic bacterial shapes or morphologies. The spherical shaped bacteria are known as cocci. These happen to be in a chain, but an individual one would be a single coccus. Bacilli are the rod-shaped bacteria, and here you see two rods. And then the Spirilla are the spiral-shaped bacteria. They have multiple turns. They're rod-shaped, but they have twists and turns in the rod. There are basically two types of stains. The simple stain, composed of one dye, which will stain all cells the same color. A simple stain will let you see cellular morphology. The differential stain has two or more dyes. You will be able to see cellular morphology, but you will also be able to put the cells into some sort of category. In doing a simple stain, you're simply going to have dyes stick to the cell. All the cells will be whatever color the dye was used. So you may have blue ones if you use methylene blue, purple ones if you use crystal violet, or pink or red ones if you use safranin. All three of these are stains that can be used as simple stains. To do a simple stain, you first obtain a heat-fixed smear. It's a good idea to cover the label with tape. The alcohol and the dyes will tend to wash off pretty much any marking you've put on your slide. While the tape won't completely protect this, it will minimize your loss of writing. Even after you finish staining, you can probably tell what was on the slide. You simply flood the slide with stain. Make sure the whole slide is covered. And you allow that to sit for 1 minute, and then you rinse with water. You blot the slide with either a paper towel or some bibulous paper, and you remove the tape covering the label. You may need to write what was on the label so you can still tell what it is. You're now ready to examine with your microscope. Remember to put the slide on the stage with maximum working distance, then minimize your working distance and start on low power or the 10x power. Use your coarse and fine adjustment knobs to get something in focus. Make sure something is in the center field that you can focus on. You then rotate the high power objective in place and use only the fine focus knob to sharpen the focus. You're now ready to move to the oil immersion lens. You want to rotate the objectives halfway between the 40x and the 100x objective so that you can put a drop of oil on the slide. Then rotate the oil immersion objective in place so that it is sitting in that drop of oil. Only the fine focus knob is used. As you have gone up in magnification, you probably have had to adjust the light with the iris diaphragm. If you haven't, probably by the time you get to oil immersion, the background is going to be pretty dim, and you may find opening up the iris diaphragm will help you see things a little better. You can note your cellular morphology at this point. The Gram stain has 4 different reagents to it. It is a differential stain, so it needs 1 or more dyes involved. The stain reaction of the Gram stain is based on properties of the cell wall. So you'll be able to categorize cells as either Gram positive, they will be blue or purple in color when you finish, or Gram negative, they will be red or pink in color when you finish. The 4 Gram stain reagents are crystal violet, Gram's iodine, the decolorizer, and safranin. To start the Gram stain procedure, you again obtain a heat-fixed smear and cover the label with tape. You flood the slide with crystal violet and allow that to sit for 1 minute. Notice how the slide is completely covered with the crystal violet. Crystal violet is your primary stain. All the cells would be purple if you stop the staining at this point because crystal violet can also be a simple stain. After 1 minute, you want to tilt the slide and rinse with tap water. You are then ready to add the Gram's iodine solution. Now you want to sort of chase the water off the slide, so keep the slide tilted while you drip 2 or 3 drops of Gram's iodine on the slide to sort of wash the water off, then lay the slide flat and cover it with Gram's iodine. This sits on the slide for 1 minute as well. Graham's iodine is the mordant for the Graham stain. It will penetrate the cell wall and react with the crystal violet to form a large blue complex molecule that's not easily removed from the cell. Again, if we were to stop the staining process at this point, all the cells would appear purple or blue in color. After a minute, tilt the slide and rinse with tap water. Now this next step is very tricky. This is a step that I can't really tell you a time for. You're going to be using the acetone alcohol solution, which is the decolorizer. Gram-negative cells have a large lipopolysaccharide component in their cell wall, and this decolorizer solution will dissolve the lipids, washing large holes in the gram-negative cell wall, allowing that big purple complex to wash out. Gram-positive cells don't have nearly as much lipopolysaccharide in their cell walls, almost none, so they aren't affected by this process. The reason we can't time this one is because it's going to depend somewhat on the cells you have and the thickness of your smear. What you want to do is tilt the slide and drip the acetone alcohol on the slide, drop by drop, until you stop having purple run from the smear. Then you immediately rinse the slide with water to stop the decolorizing process. At this point, if we were to stop the staining process and look, the gram-positive cells still have retained that blue complex, but the gram-negative cells are now colorless. So you're going to flood the slide with safranin and stain it for about 30 seconds. Then you tilt the slide and rinse with tap water. Safranin is the counter stain. Since your gram-positive cells are already stained, they will not pick up any of the safranin, but the colorless gram-negative cells will take on the pink safranin. This will leave your gram-negative cells pink or red, or the gram-positive cells blue or purple. Once again, you want to blot the slide with a paper towel or bibulous paper. Remove the tape covering the label and probably refresh the label a little bit. Then you can examine with the microscope. Again, put the slide on the stage with the maximum working distance, rotate the 10x objective in place, and minimize your working distance. Focus using the coarse and fine adjustment knobs. Put something in the center field that you can focus on. Then move to your high power. Use your fine focus. Rotate halfway between the 40x and the 100x so you can put that drop of oil on the slide, and rotate the oil immersion objective in place. Only use your fine focus. Again, remember to adjust light with the iris diaphragm. This time you can note cellular morphology and the color of the cells because now you'll have a Gram stain reaction. When you report a differential stain, there are three things you need to put in your report. You need to tell what type of stain it was. In this case, it's the Gram stain. The stain reaction, whether it's positive or negative, and your cellular morphology. Did you see cocci, bacilli, or spirilla? If you have blue spherical cells, these are reported as gram-positive cocci, or GPC. Pink spherical cells would be gram-negative cocci, or GNC. Blue rod-shaped cells would be gram-positive bacilli, or GPB, and pink rod-shaped cells would be gram-negative bacilli, or GNB. The spirilla of medical importance do not stain well, so we don't have any kind of standard abbreviation for them. Usually, these cells are demonstrated with dark field microscopy. Now you may get gram-variable reactions. That is, you'll have both gram-positive and gram-negative cells on your smear when you know you took your smear from a pure culture. There are two reasons for this. If the culture is old, there may have been some cell wall deterioration in the older cells, and they may not have retained the dye as they should have. The other reason, particularly people who are just learning to gram stain, is you use too much decolorizer.